Good morning and welcome to this webcast. We're glad you're joining us here for this final STEMnet webcast of 2020. Hello there, I'm your host Jennifer Wicks behind the scenes. Glad to have you on board for this special webcast, Exemplars in Biology. We will have captions throughout the session, as you can see in this webcam or this web pod beneath me here. Please do engage through this session. We'd love to hear from you with your questions and comments over in the chat pod that will be on screen through the entire session. We will have the Q&A primarily at the end today, so please do stay tuned for that. And with that, it is my great privilege and pleasure to work with the STEMnet team and introduce our Executive Director of STEMnet, Dr. Frank A. Gomez. And with that, I will go ahead and hand it over to you, Dr. Gomez, to get started. Thank you.
Great. Thank you, Jennifer, for your kind introduction, and welcome all of you to our final webcast of the year entitled CSU Exemplars in Biology. I hope you and all your families are all healthy and safe during this holiday season. Stellar research by our biology faculty and students abounds at CSU, and we would be doing an injustice if we were to focus solely on any one subdiscipline of biology. Hence today we're highlighting but a few examples of some of this work to demonstrate what is possible if one shoots for the stars and provides experiential opportunities for students, sometimes beginning quite early in their undergraduate education. It is here in the CSU, more so than any other system of higher education in the country, and with such a diverse population, that we offer students access to learn by doing, allowing them to see firsthand the marvels of research and putting what they learn in the classroom into action. Today's exemplars are proven champions in their respective biological fields. I might also add that they're currently funded by either NSF or NIH, including an NSF Career Award recipient. So before we begin, I want to remind the audience to use the open chat box on their screens to type in their questions. Our speakers, when not presenting, will make every effort to respond in real time. So let's begin and hear about some of the exciting work happening in the CSU and from your colleagues. Our first speaker is Scott Roy from San Francisco State University. Scott, the floor is yours.
Great, thank you, and thanks to the organizers, and thanks for everybody for joining us. Good morning. I'm going to be talking to you today about our work, which is on the origins of diversity of introns and other bizarre genetic phenomena, sorry, I'm working with the tech here. Oh, it did work. As I like to think about it more concisely, just weird genetics. So just a little bit about me. I've been at San Francisco State since 2012 after doing a series of fun postdocs all over the world. I teach genetics and bioinformatics, and I run an all bioinformatics dry lab. One thing about our work is we collaborate a lot with a bunch of different colleagues from SFSU and beyond. But mostly I wanted to just put up my CV so I could have an objective, which is to give today's weirdest talk. So my history with weird genetics starts when I was an undergraduate, a classic sort of the importance of an undergraduate research experience, which was in the introns early debate. So to remind you, introns are these bits of DNA that interrupt genes and then need to be removed from RNAs before proteins are translated. And so the introns early hypothesis said, well, why are there introns? Maybe these are the remnants of creation of the first genes. And so the hypothesis said maybe we started out with small DNAs encoding small RNAs encoding small proteins that by occasional chance events got combined together to give the modern intron-exon structures that we see. And so at age 20, I was told, hey kid, basically, can you see if there's a correlation between intron-exon structures and protein structures? If there is, you will have explained the origins of the first genes. And of course I was hooked, even though I think we were wrong. And now I've continued to sort of work on the strangest things I can find. So one is genome scrambling in ciliates, in which this organism has both germline and somatic genomes, but the somatic genome is produced by all these crazy rearrangements of the germline genome. Another thing that we're working on is paternal genome elimination, which is found in some insects. So whereas females do the normal thing, they get paternal chromosomes and maternal chromosomes, but then when they undergo meiosis, they scramble them up to make a diversity of gametes. But males discard their paternally inherited chromosomes and transmit only their maternal ones, going to sort of in the extreme haplodiploidy, where there's not even sperm involved in production of males. So when we address these questions, we want to basically think about what is going on, how is it done, and why might this have evolved. So I want to tell you two stories today, one about introns and one about sex chromosomes. So the story about introns basically tries to explain this phenomenon where across eukaryotes, this is a diversity of eukaryotes, you have some lineages that have many introns per gene, and then you have other lineages that have hardly any. And so the hypotheses for these have been, well, maybe complex organisms need more introns, maybe species that replicate rapidly can't tolerate introns, or maybe even selection is differentially efficient in different lineages. So this is basically a history of intron loss and gain. Those species with lots of introns have to have either gained more or lost fewer. So we've been trying to find intron losses and gains. So the first attempt was comparing mouse and humans. And what you see here in the bottom left is we aligned protein sequences from mouse and humans and fish and marked their intron positions. Quite to our surprise, we found that there are pretty much no changes, indicating that there's been neither loss nor gain in basically the whole history of vertebrates. However, as we continued to look across species, we did start to find some that show a different pattern. So this is from a brown alga, and what we see is this really distinctive pattern where there are a whole bunch of introns that are of the same length. And when we look at those specific introns, what we find is that they basically have all the same sequences, which indicates that they were created by insertion of transposable elements that somehow have evolved the ability to create introns, which we call introners. So that gives a sense of how introns are created, which allows us to then turn to the question of why are some lineages creating introns and others not. So this is work I did with an undergraduate, Landon Gozacci. So we searched for introners, for the signature of similar introns, across 3,000 eukaryotic genomes. So we looked across this whole diversity of organisms. I've highlighted plants and animals, all plants and all animals, to give you a sense of how much diversity we're talking about. In asterisks, I've shown the lineages where we did find introners. Out of all these species, we found only a fairly small number of lineages that have found them, about 100 species. And so then we dug in to try to figure out why. And what we noticed was that most all of these species are lineages where we have reason to know or to suspect that there's been a lot of lateral gene transfer, acquisition of DNA from the environment. So quite different from the hypotheses that have been put forward. What we think is going on is those lineages that acquire DNA from the environment sometimes acquire introners, which then go on to spread introns and give high intron numbers. That it might be lateral gene transfer that is key to explaining gene complexity. The other story I want to tell you about is about the strangest known mammalian sex chromosome. Nearly all mammals have the familiar XY system that we inherit from a deep mammal ancestor. However, there are occasional exceptions. Perhaps the weirdest one is Microtis oregoni, or the creeping vole. And to understand this, we can start with noting that the XY males lose the X chromosome during spermatogenesis, producing either Y-bearing or sex chromosome lacking sperm. This then has implications for female production, because females are produced from non-Y-bearing sperm, leading to what we call XO females, females with a single X. However, they do the opposite thing now, and the X chromosome drives so that all eggs contain an X. So we wanted to understand what we could about this weird system, so we went in and sequenced their genome. And so the first thing that we noted was the following pattern. So in general, when you sequence a biological sample, the amount of sequence that you get corresponds to the copy number. So if you look at a female, you see basically the same amount of the same density of reads for X chromosomes as for autosomes, because you have 2 X's for every 2 autosomes. But in males, in most species, you see a pattern where you see only half as much X chromosome sequence. So when we looked at our species, we first looked at females and found, as expected, that there's evidence for only 1 X in 2 different females. However, to our surprise, when we looked at males, we found that there were actually 2 X's for each 2 autosomes. So when we went in and actually sequenced the genome, what we found is that the X chromosome is basically a normal X chromosome. However, the Y represents a fusion between the X and the Y. Sorry, there's a little error here. But basically that the crouping vole, so-called Y, is actually a fusion between the X and the Y. So what's going on here? Well, we think this could be a response to so-called feminizing X chromosomes. These are known from many rodent species, where you have this extra X chromosome, a weird X chromosome that causes XY females to develop, XY individuals to develop as females. We think that these weird X chromosomes gain an advantage when you have a cross between XY females and XY males, because you produce YY offspring that perish, because everybody needs to have an X chromosome, because X's are like X's, everyone has one. I'll assume everybody's laughing, muted. So that basically you get the X chromosome gains an advantage. So then our hypothesis would be that the Y chromosome that could acquire the X-linked genes would benefit because they would no longer perish and therefore have higher fitness. So we're still working on this, as with both stories, so stay tuned. So that's what I wanted to talk about our science, and we've also been asked to talk a little bit about what we've learned as CSU biologists. And so I have three points I'd like to make. One is just to keep trying along a whole bunch of axes. So we work with a variety of students, a lot of junior students who are just getting started in research, and I find that really challenging. So I'm not going to sit here and tell you that I figured it out. I keep trying to learn from the people around me, trial and error. A couple of things that that seem to be working are co-mentoring, as well as working with student teams. Just trying to would be just keep sending the grant back in. I finally got funded on my third try. I didn't even edit it from the second one, the second draft. I just sent it in again, and this time it got funded to the same panel. So just always remember that grant proposals they don't have to be your best work. They are a lottery ticket. Give yourself a try. Keep trying 3 is to look for funding instruments that are interested in you. I have gotten some insulting reviews from NIH and NASA, but NSF seems to like me, so just keep at it. And a fourth is a story about a colleague, one of my colleagues, who in the same semester, as he got rejected by C-Superb, he got a perfect score on his NIH grant. So don't be dejected, just keep at it, is the only advice I can give on that. A second key thing, I think, is to assess and use your strengths. So for me, bioinformatics is a strength because it's cheap, but it can be a liability because I don't have really entry-level tasks for students, so I can struggle to get students that first enter the lab involved. Similarly with junior students, we all know the joys and importance of mentoring junior students, but it can be one can struggle to complete projects when was working with people who are having their first research experiences. Having a niche is important because it's hard to compete with big institutions at a CSU, but on the other hand, sometimes my students can struggle to appreciate the importance of some of the questions I'm interested in. And collaborations are great, but I've tended to have too much on my plate. And so to me, it's been really important to sort of assess and reassess my strengths. You can see that in sort of what's worked in my grant. My grant project is basically the obvious thing, what you would imagine from 15 years ago that I would be doing. Not a whole lot of creativity, just an obvious, apparently, hopefully important thing to do, and I'm the one to do it. I proposed a cure in my bioinformatics class, which the NSF really liked, and I've used the advantage of bioinformatics to involve more students and to work with community college students. And so that is me in summary. I told you about 2 projects, one on how introns and why introns are gained, one on bizarre sex chromosomal systems, and some notes on what has worked for me in my lab. So thanks for your attention. I'm looking forward to everyone else's talk. Next up is Kelly Young from Cal State Long Beach. Take it away, Kelly.
Thanks, Scott. Hi, everybody. I'm really excited to share with you some of the work that my undergraduate researchers have done in the Briefer Lab here at CSULB. And let's see, the data set that I'm going to share with you today is a really simple one. And that's because, as Scott said and Frank indicated as well, we were asked to share some tips about working with CSU students in the lab. And this data set arose actually because of working with undergraduates in the laboratory. So in our lab, we work with seasonal breeders, and individuals with seasonally breeding species are simply individuals that can limit their reproductive function to particular times of the year. And this is going to enable them to optimize survival for both parent and offspring. Now, these individuals are able to time their reproduction because they can both assess or they can measure and respond to changes in photoperiod, and that's just the number of hours of light in the day. Now, photoperiod affects reproduction through the HPG axis, or hypothalamic pituitary gonadal axis. And what this means is during the breeding season, and that's shown on the left side of the figure, stimulatory photoperiods are going to induce the HPG axis to produce high levels of GnRH from the hypothalamus, high levels of gonadotropins from the pituitary. These are the follicle stimulating hormone and luteinizing hormone. And with high levels of FSH and LH, you see an active ovary. Now, in contrast, in the non-breeding season, we see depression of the HPG axis. So inhibitory photoperiods are going to reduce GnRH, reduce FSH and LH. And what we see in the ovary is a reduction in the number of mature or antral follicles, a reduction in the in ovulation, low estradiol production. Essentially, everything that the ovary is supposed to do, it stops doing in terms of reproductive function. In the literature, the ovary in the non-breeding season has been termed quiescent or inert, atrophied, non-functional. And in our lab, we're really interested in that transition between the non-breeding season gonad and the breeding season gonad, and how and what it takes to get a turned off ovary to turn back on. Now, we are able to manipulate our seasonally breeding species in the lab by just changing the photoperiod that they're exposed to. And the model that I'm going to talk about today are Siberian hamsters, or Photophis angoras. And Siberian hamsters are long day breeders. And this means that they respond to long days for breeding function, or summer type days. And for us, a long day is 16 hours of light per day, 8 hours of dark. When they do that, what you see is in that top panel is an ovary that's functional. There are some antral follicles there. Those are the mature follicles, and there's corpora lutea evident as well. Those are the structures that form after ovulation. That's an active ovary. Now, in contrast, if we were to place a Siberian hamster female in short days, and for us that's 8 hours of light per day, 16 hours of dark, we see a regression or a shutdown of ovarian function. So no mature follicles, no ovulation. Now, the exciting group for us are our experimental groups. These are our recrudescing ovaries, or returning back to function. And to get these particular ovaries, we transfer females into short days for 14 weeks, inducing regression, and then transfer them back into long days, anywhere from, we've done 2 days up to 8 weeks. And these are our post-transfer, or PT groups. And today I'm going to talk about data from post-transfer week 2, 4, and 8. Now, every time we expose females to these particular photoperiod regimes, we see the same results. Now, all the graphs that I'm going to share today have the same x-axes. So if we look there, LD is the long day or the cycling ovary, short day FD, that's the regressed female, and then our post-transfer returning back to function weeks 2, 4, and 8. And these graphs indicate mean plasma FSH, mean plasma estradiol, ovarian mass, paired ovarian mass, and paired uterine horn mass. And again, we see the same pattern every time on these reproductive parameters, which is a U-shaped pattern where levels are high in the long day active ovaries, low in short days, and returning back to levels above short days, or returning back to long day function in our post-transfer group. Just what you would expect. Now, every time we run an experiment in the lab, we replicate these data. We have students do obviously the masses, but they do the hormone assays again, and they do histology every single time. And we do this, even though this has been published again and again. Now it's sort of an aside, like, okay, we see the same results. But because for our lab, and I know Scott indicated that it's not always possible in every lab, but we have some techniques that are more approachable. And I always recommend starting with something that is a little bit more approachable, particularly for new undergraduates. And for us, that's histology. So everybody knows you come to the lab, you're going to get an ovary, you're going to embed it in wax, cut it on a microtome, stain it, and then count follicles. And it's great to provide confidence for students before they move on to something a little more tricky, like working with RNA. Now, like I said, we do this with every single experiment. So undergrads have counted a lot of follicles in the ovary. And again, we see the same results. Here, these are graphs showing tertiary follicles. Those are our mature follicles, and corpora lutea, the structures that form after ovulation. That same U-shaped pattern, so high in long days, low in short days, and returning back to function in our post-transfer group. but I have students not just count the mature follicles, the ones that are directly leading to ovulation or indicating ovulation, but every follicle. Because in the ovary, the process of folliculogenesis starts with primordial follicles. These are immature follicles that mature over time into primary and then secondary, but still these earlier stages are really thought of as immature follicles. They can't even respond to FSH. So with these earlier stages, what we've seen again and again and again was no change. And this is something that we really had as an aside for so many years. It's okay, those early follicles, they never change. But it wasn't until a student actually said, we were talking to students, and well, why don't they change? That ovary is actually regressed. Why aren't they changing? That it led us to this hypothesis of, or a question of, well, maybe that shutdown ovary isn't fully regressed. And that led us to a testable hypothesis that maybe processes dependent on FSH will change with photoperiod, whereas processes independent of FSH won't. Because mature follicles can respond to FSH and those immature follicles up to early secondary don't. So in the lab, students always work in teams, and so everybody gets their own piece of the project. They get their own genes set, and that's been very, very helpful. So they work together, but they have their own, they have ownership of their own parts. And so we have students, this is just real-time PCR data, mRNA expression looking at FSH receptor, insulin-like growth factor 1, cyclooxygenase 2. These are factors dependent on FSH. And they're there in the part of folliculogenesis for mature follicles and for ovulation. That's where they're important. And we see our same U-shaped curve, high and long day, low and short day returning. However, when we look at factors that are important in those early stages of folliculogenesis, the transition between primordial and primary and secondary, these are transcription factors and growth factors, we saw no change. This is very similar to what we saw for those early follicle counts. Now, for the student who was actually doing this PCR, it was a little depressing to continue to get no difference, no difference, no difference. But for us, it was really exciting as we started to talk about what this means. And again, it's a simple premise. Perhaps the ovary in the non-breeding season isn't inert. Perhaps what we're seeing is folliculogenesis continuing up until the point where it can't. Remember, there's no or low FSH on board in the non-breeding season. So the ovary maybe just be functioning up until the point of where it just can't proceed because there's no FSH and those later stages are dependent on FSH. A simple story and a simple conclusion, but for decades, the way that we've referred to the non-breeding season ovary is an organ that's non-functional, inert, atrophied, and really it's still going. And this has some important implications for both agriculture and also clinical studies where we see ovulation not happening, but those earlier stages still going. Now, in case you're interested, FSH isn't the only factor. We've done some fun whole ovarian culture looking at adding different growth factors important for recrudescence. We're finishing up an FSH ablation replacement experiment, and RNA-seq data from a recent paper showed lots of patterns, not just those that U-shaped pattern, but lots of different patterns of gene expression. So there's more going on here than just ovaries turning on and turning off. In terms of lessons learned for me, really encouraging scientists to ask simple questions. And I know we all do this, but for me, it was that change of just encouraging them to ask questions, but then actually following up on it and saying, all right, let's test that, even if it wasn't directly in the grant. I really believe in having students, when possible, start on those easier techniques. It gives them so much confidence in the lab. They're doing a lot of hands-on work, but then they actually succeed more when they start doing trickier techniques. And I also am an advocate for pairing students in teams where they have their own piece. They're dependent on, they've got their own genes that they're responsible for. Even though they're working cooperatively as a whole, they're not totally dependent on somebody else for the other half of the puzzle. They've got their own little piece of data. Finally, for me, one of the things that I've noticed in the last few years is the more that I reflect on how I mentor and to be more mindful about my mentoring has really made a difference. And I'm so taken by this. I really am all in for mentoring and learning about mentoring that I'm a part of our CSULB Build initiative where we're creating mentor training programs. And we've created 2. They're free to CSU. So if you're interested in bringing a formal mentor training program to your campus, let me know. And also, I'm just really excited for what our students can do in a CSU laboratory. I am a huge advocate for undergraduate research. Get them started thinking about ways to get them involved. What are some simple things within your field that you can get them involved with, because they can do the work and they can present, they can be co-authors and all of that. Right now we're working on hypoxia in the ovary. We're working on the ovarian stroma, not just the follicles, beta adrenergic stimulation, and retinoic acid signaling in the ovaries. So we've got a lot of different things going on with undergraduate researchers. So with that, I want to thank my lab. Don't worry, this is a 2019 picture. We didn't get a 2020 picture with our masks. And also NIH for its continuous and awesome support of undergraduate research at the CSU. So thank you. And with that, I'd like to introduce my colleague and build colleague also, Dr. Gilberto Flores at California State University, Northridge.
Thank you, Kelly. Can you guys all hear me okay? We had a technical issue and I just want to make sure my mic is okay. So, all right, it looks like you guys can hear me. All right, well, thank you, Kelly, for the introduction, and thank you all for joining me, and also thanks to STEMnet for giving me the opportunity to share with you guys our research on the genomic and functional diversity of the human gut symbiont, Akkermansia muciniphila. So I'd like to start with a little bit of introduction. As most of you are probably aware, the human gut microbiome is really an emerging field of research in human medicine. And to define the microbiome, it's really the collection of microorganisms that live in the large intestine in terms of the gut. And a lot of the early work in microbiology was focused on the pathogenic organisms that live associated with humans. But what's really changed in the last 10, 20 years is that we started looking at the whole spectrum of symbiosis. So the mutualistic organisms as well as commensal organisms. And it's really with these mutualistic and commensal organisms that we're starting to appreciate how they perform essential functions really throughout life, whether it's for digestion, inhibit pathogen colonization, as well as educating our immune system. And some of the more recent work that's coming out is really exciting, looking at mental health and behavior in humans. And what's really emerged from these studies is that we can start to link changes in the composition of these communities, and really the term dysbiosis is what is used to kind of describe this change in these communities. They are associated with several diseases, and we do need to be careful because most of these are associations, and so it's really difficult to demonstrate causal relationships. And so that's kind of where the field is heading. And as a microbial ecologist, I would argue that for us to really exploit the microbiome to promote health, we need an understanding of the ecology of the system, and this really spans the spectrum of biological organization from community, population, species, and strains. And so what's also come out of this is really the pursuit and kind of the identifying of potentially therapeutic microorganisms. And so one of the emerging therapeutic microorganisms is our bug of choice, I guess you could say, in our lab, and that is Akkermansia muciniphila. So this is a member of the Verrucomicrobia phyla, which is a relatively underexplored phyla compared to plenty of the other bacterial phyla. This organism was first isolated in 2004 by a group in the Netherlands, and since then there's been a number of studies showing that it's present throughout life, so in infants, and then even later throughout life, even into the old age. Typically when it's present, and so it is present in about 70% of the people whose microbiomes have been looked at, it's usually there at about 1 to 4% relative abundance. And as the name implies, muciniphila, it is mucin loving. And so it uses mucin, which is a glycoprotein that lines our epithelial cells and really is the main structural component of mucus. And so it uses that mucin as a carbon and energy source. And the role of the mucin and the mucus layer is really to protect our epithelial cells. So intuitively, you might think that it's not necessarily a good thing that it's eating the mucus barrier because it is protecting us from other microbes. But what happens is that when it eats the mucus, it actually produces acetate as one of the main fermentation products, and that acetate fuels our goblet cells, which are the ones that make the mucus in the large intestine. So you can see in this diagram here from this work that was done in 2013. Here along the x-axis we have mice that were on a chow diet, control diet. administered Akkermansia, then a high-fat diet, which we know erodes the mucus barrier. And so you can see if we just look at control, high fat, we see a depletion in the mucus thickness. But if you administer Akkermansia with the same high-fat diet, it does restore that mucus thickness. And so what's going on is a positive feedback. Again, that acetate is fueling the goblet cells to make more mucin. There's many other benefits that have been explored, including improvements to metabolic profiles. There's been some recent work showing that people respond better to certain cancer immunotherapies if they have Akkermansia, and there's some ideas about priming of the immune system. There's even some work that was done down at UCLA showing how if you put people on a ketogenic diet, it reduces seizures, and it seems to be related to the microbiome, and in combination with another organism, Akkermansia seems to be part of the role of that. And all of this has led to a clinical trial that was done last year in Europe, evaluating the safety and efficacy. And I put these are two companies that have emerged, one in Europe, Akkermansia, and one here in the US, that are really pursuing Akkermansia as a therapeutic for a variety of different diseases. And so what really struck me when I started getting into this work was that everything we know about Akkermansia and all of these benefits are based off of one described species. And so if you're a microbiologist, you would think that that is actually rather dangerous, because we know that strain level diversity, species level diversity is very important. For example, we probably all have E. coli in our guts right now, but we don't have the pathogenic version of E. coli, right? So it's really dangerous to assume that all of them are behaving the same. So despite the known genetic diversity, so here's a tree from a group out of China in 2017. They were able to isolate 40 different human-associated as well as mouse-associated Akkermansia. And you can see in this tree that there is diversity within the Akkermansia lineage. And so they were able to define 3 species-level phylogroups. And so we can think of a phylogroup in this case as a different species of Akkermansia. The type strain, this one up here, ATCC BA335, that is represented in the group 1 phylogroup up here. I'm just figuring out this pointer, so it's new. And the other thing that they noticed is that there was an open pangenome across the Akkermansia lineage, which means that there is unknown diversity that is yet to be explored. And so about the same time, we were awarded an NIH SC2 grant to explore the genomic, metabolic, and cultivable diversity of human-associated Akkermansia. And really this is operating under the hypothesis that this functional diversity of Akkermansia really influences how they impact human health. And so I had a data set of metagenomic sequence data, and for those of you that aren't familiar, metagenome is the sequencing of the entire microbial community's DNA. So we can get not just who's there, but we can also get an idea of the functional potential of these communities. And so we had these data from 75 children, and from that we were able to reconstruct 36 metagenome assembled genomes. So this is essentially putting genomes of Akkermansia together. And we're fortunate because there's not a lot of these verrucomicrobi in the gut, and so the genomes of Akkermansia are very distinct. And so we got really good assemblies, we got really good genomes. They're not closed, but they still are high quality genomes. We combined those 36 with the 39 from that previous phylogenetic tree from the group out of China, and we used comparative genomics to kind of predict functional differences amongst these phylogroups. And so here are some of our results. I know there's a lot going on here, but I want to point out that in this circular diagram, each of these concentric circles is a bacterial genome. And so moving out, we have 75 total genomes. And we can see here in purple, blue, and then orange that we have purple, blue, and orange. We have the 3 phylogroups that the group out of China was able to define. We were able to identify a 4th phylogroup here. This heat map is showing average nucleotide identity of each pairwise comparison of each genome, and so we were able to define a 4th phylogroup, the AM4. We also were able to identify functional differences. So we can see in this region we have the core genes that are shared. We have these singleton genes, but really where it starts to get interesting is up here, where we have these patterns of genes that are present in 1 or 2 phylogroups and then absent in the other phylogroups. And so blue represents our AM2, orange is our AM3, and what we were able to identify here was the presence of these genes coding for core and ring biosynthesis. And so this is involved in biosynthesis of vitamin B12. So if we take a closer look, we can see these are this core and ring biosynthesis gene cluster. We can see that it is absent in the AM1, present in almost all of the AM2 and all of the AM3, and then also absent in the AM4. And so at the same time, we were also isolating and building our own culture collection of Akkermansia. And so here's a phylogenetic tree showing our isolates that we have here in the lab. At the time, we didn't have our group 3, but we now have a group 3. But you can see here we have the group 1s of the type strain, which are classified as vitamin B12 salvagers, and our producers, AM2 and AM3. We were able to verify this. I don't have time to go into the details, but we published this earlier this year, but we did a bunch of experiments to verify that these were producing vitamin B12. And so why this is important is that if the bacteria produce vitamin B12, they can make propionate. And propionate is one of the fermentation products that Akkermansia makes. And it's anti-inflammatory, and it also helps to regulate our appetite. So we're not as hungry when propionate is being produced. More broadly, there's a lot of interest in vitamin B12 in multiple microbial communities because it seems to be an important modulator of microbial ecology. As an example, 86% of the microbes whose genomes have been sequenced, bacteria whose genomes have been sequenced, require vitamin B12, but only something like 36% of them actually make vitamin B12. So it seems to be a community good that is exchanged and facilitates a lot of interactions in microbial communities, including the human gut. So we also noticed a bunch of other functional differences, and this kind of led to our next grant. So this year we just received funding in SC1 from the NIH to look at the mechanisms and consequences of human milk oligosaccharide growth and bile stress across diverse strains of these Akkermansia. And the reason this is important is that both HMOs, human milk oligosaccharides, and bile really impact the timing and success of colonization of bacteria in the human gut. I don't have any time to go into any of these results, but we are working on this area. So if you have any interest in human milk and bile in the microbiome, I'd be interested in talking with you. So in summary, human-associated Akkermansia are genomically and functionally diverse. And I would argue that it is premature to bring to the market as a therapeutic. As I mentioned, most of us already have some version of Akkermansia, so it might also be more prudent to stimulate the ones you may already have. More broadly, I think this is an example of how we can merge these cultivation independent, so basically these sequencing approaches with cultivation dependent approaches in microbial ecology. And so for my last minute here, I just want to talk briefly about this idea of building on the success of the CSU. I think some words of advice, and like Scott had mentioned, I'm by no means an expert, and I'm continually learning and developing new strategies. But I think what's important is to be strategic. There are specific programs for us, and we do have strengths in terms of our ability to do cure style courses. These play well at the NSF, and I also think building the right team is important. When writing the grant, I think we all try, but you have to be thorough. You have to think like a reviewer. If you get caught up on something, or if one of your colleagues who's reading it, the reviewers will get caught up on it as well. My first grant at the NIH was a pilot grant, but preliminary data, even in a pilot grant, I think is extremely important. And one of the comments I often get in reviews is feasibility. There's still places that don't think we can do certain things at the CSU. And so if you can show them that you're already doing it, it's going to really help your chances. As somebody already mentioned, Scott mentioned, talk with program officers, before and after submission. I think that's also important. And I also learned from my colleagues here. Mike Summers recently retired, but I kind of looked at the way he had built a successful research environment at CSUN, and I really learned from that in terms of scope of projects, the type of stuff that works in this environment, and then also if they have past reviews to see what kind of people get hung up on. And so I'd be happy to talk more, but I am out of time. And so with that, I would like to introduce Pleuni Pennings from San Francisco State University. And thank you for your time.
Thank you, Gilberto. Hi, everyone. I see that I look a little fuzzy, I think. It's dark here. My name is Pleuni Pennings, and I'm normally at San Francisco State University, but this semester I'm in Amsterdam for a sabbatical. So it's evening here, but I know for most of you it's morning, so good morning. I'm going to be talking today about fitness cost of mutations in viruses. And mutations in viruses are very common, and they're very important for viruses, and they can be dangerous for us because mutations in viruses can make viruses become resistant to drugs, for example, or they can become more virulent or more pathogenic. And so typically we are worried about mutations in viruses. But many mutations in viruses actually are bad for the viruses. They come with a cost for the viruses. And we are interested in studying these costs because we believe that if you understand basically what breaks the virus, like which mutations really mess up the virus, that can help us learn how the virus works. So we can learn something about the function of different parts of the viral genome. So this talk is about an NSF-funded project, which is about fitness costs of mutations and diversity in viruses. And it's a collaboration with two other labs, Dr. Adi Stern's lab in Tel Aviv University and Dr. Bob Schaefer at Stanford. And I think our most important discovery until now is that CpG sites are very costly for HIV, but surprisingly not for hepatitis C virus. CpG sites basically just means that there's a C next to a G in the genome. And so you would think, since there are only 4 letters, all of them are going to be each other's neighbor sometimes. But a C neighboring a G is actually bad for viruses. It's actually also bad for us mammals. But for viruses, it's bad because these CG sites interact with our immune system. And so our immune system recognizes viruses in part by their CG sites. But for some reason, there's a difference between the effect in HIV and hepatitis C. We don't know yet why that's the case. The highlight of the last couple of years with this grant has been by far our trip to Tel Aviv with the entire lab. I took 9 students on a week-long trip to Tel Aviv to go to the lab over there to meet all the students and our collaborators, to present their work, to hear from the work that was being done in Tel Aviv, and also to see the city. And it was absolutely wonderful. And I wasn't sure if it was going to work to take some of these students were very young undergraduates, and I wasn't sure if they were going to actually benefit from this kind of exposure. But it was absolutely wonderful to bring them together with students in Tel Aviv. Actually, there's a lot in the grant that we have not done that we plan to do, that we're still doing now. But instead, we've done other things. We've studied many viruses, worked on a big hepatitis C data set, even though the grant was for HIV work only. We published quite a few papers out of this grant. This is the most recent one that was published in PLOS Pathogens. It's work on several viruses, mostly data from Tel Aviv. This is a paper, is a short commentary actually, but I thought it was fun to mention because it's about SARS-CoV-2. So since I published a little bit about CpG sites, I got involved in sort of the little world of people who work on CpG sites and viruses. And someone published a study saying that because of some patterns in the CpG sites in SARS-CoV-2, they could see that SARS-CoV-2 came from feral dogs. And then several people independently of each other said that was nonsense. And so many of us wrote comments or talked to journalists about that paper saying that it was nonsense what they were saying. And then we all found each other and wrote a commentary together, basically saying that other paper was nonsense. One of the things that I do in my lab and that I have my students do is I make short videos about research and about other things. I've made some videos about COVID that have been extremely successful. One of the videos that I made last year about R0 and flattening the curve has been watched 5 million times. Most of my videos do not get that kind of exposure, but even if just a few hundred people see it, it makes me very happy. So I'm going to show you a short version of a video about one of the papers that came out of this grant, and the video and the paper is all done by students.
In this video, we will tell you about our work to determine whether CpG-creating mutations are costly to human viruses. It is based on work done by students from San Francisco State University, Victoria Quado, Serena Quinn, Ryan Winstead, and me, Jasmine Carr.
We looked at different viruses like dengue, influenza, entero, herpes, hepatitis B and C, and polio. So, what is a CpG site? When we are looking at a genome, a CpG site is when you see a cytosine nucleotide followed by a guanine nucleotide. Now we will explain how a CpG site is created. One example is when a C precedes an A and the A mutates to a G. This point mutation creates a new CpG site. In our paper, we compare mutations that create CpG sites with mutations that do not create CpG sites.
So what did we find out in our research? Since we analyzed over 100,000 sequences, we will explain only a small portion of our findings. Here we will focus on synonymous A to G mutations for dengue 1 virus. We show here the frequency of every synonymous A to G mutation in dengue 1. On the left are the mutations that don't create CpG sites, and on the right are the ones that do create CpG sites. You can see here that non-CpG creating mutations have higher frequencies than CpG creating mutations. When tested, we found that this difference was significant. This result suggests that CpG forming mutations are more costly than the non-CpG forming mutations. That's why they occur at lower frequencies.
To figure out how costly CpG-forming mutations are, we graph the ratio of the average frequencies of non-CpG-forming and CpG-forming mutations. If this ratio is higher than 1, it suggests that CpG-creating mutations are more costly than non-CpG-creating mutations. The higher the ratio, the more costly the CpG-creating mutations are. The main conclusion of our paper is that, overall, CpG-creating mutations are costly for most human viruses. For more information, you can read our paper, which you can find in the link below.
Thank you. So this work was done mostly by the 4 students that you saw in the video, but there were many other students who played a small part in this project as well, and so we had a lot of student authors on the paper. One of the reasons that this project was doable for students is that we studied many different viruses, and I could assign a virus to a student or a small group of students. It was a lot of fun, and just like some of the others have said, it's not always easy to find projects that students can work on. So I was very happy that this project worked so well. Some of the lessons that I feel like I learned from this specific NSF grant is but all of these are actually also true for, I have an NIH grant as well. So we had a paper ready, I think this is related maybe to like the preliminary data that Gilberto talked about. So in our case, we had a paper on the bioarchive up before we send in our application for NSF money. And I think that really helped to show that we could do what we were saying that we were going to do. But it also helped us because it meant that we actually had already written the paper, so we had a lot of text and references already done. We had help from a grant writer. This was in part thanks to SS State, our ORSP office, but I also spent a lot of my startup actually on grant writers, and and I've been very happy with that. I think it's really helped me be successful in getting grants. My grants include money for postdocs. I find that important for the kind of work that I do, to have someone with a PhD do some of the work. And I've had quite a few postdocs. It's hard to find good postdocs who are interested in coming to a CSU. But I've had one particular postdoc, Kaho Tistammer, who's joined my lab about 2.5 years ago, and she's still there, and she's really amazing. So I feel very lucky, but it's not easy to find good people who fit at the CSU. Grant administration is not my favorite task, and I wish someone else could do that for me. So having this NSF grant was great, but actually I am really interested in working on bacteria and drug resistance, specifically in bacteria. So I'm trying to switch. I'm focusing on a new grant with new collaborators. Again, someone in Israel, Ruth Hirschberg, and Brandon Obunu from Yale were writing a grant to look at drug resistance in E. coli. Okay, the last thing that I want to share is we also have NSF-funded programs for undergraduates and graduates to learn data science and computer science. And I also run several online programs for students, both at SF State and elsewhere, to learn programming in Python and R. And I'm really excited about that, because I think that our students could really benefit from confidence in coding, in data analysis, and but it is often really hard for them to get that kind of skill. So I'm very interested in working on that kind of work, doing that kind of work and getting students these kind of skills. So with that, that was my last slide, and I would love to introduce the next speaker, Lisa Wa from Sonoma State University.
Thank you so much, Pleuni. Are you guys able to hear me all right? Hopefully, yes. Great. All of the speakers have had wonderful presentations so far, and I've actually learned so much just listening to their talks. So, hi everybody, I'm Lisa Hua, and I'm assistant professor at Sonoma State University. I actually started my lab here about a year ago, and I want to just talk about some of the projects that are really just commencing in my lab, and some of the undergraduates that have taken it on. So I'll be talking about chromosome organization in today's talk. And again, I want to thank Frank, Monica, and Jennifer, right, she's producing all this, and the STEMnet team for the opportunity to share our research across the CSU. Okay, so first, right, as you all know, homologous chromosome pairing, okay, during meiosis is this essential feature for genetic recombination and diversification, right? However, homologous chromosome pairing is harmful in mammalian somatic cells and has been correlated with genome stability and actually has been seen as a characteristic of cancer, right? So little is known how pairing of homologous chromosomes are inhibited during somatic cell generation. But what you see is that if we look at human epithelial cells, so this is just a video of human epithelial cells undergoing mitosis, right? You can see these chromosome movements are very dynamic. So this is a video coming from Alexei Kojakov's lab up in New Jersey, okay? What we've seen, though, is that recent interface data, here we go, shows that actually homologous chromosomes don't interact frequently with each other, suggesting, right, let me get my mouse out, suggesting they may be actually far apart in the nucleus. So we really hypothesize that there must be a mechanism that prevents homologous pairing in somatic cells. All right. Yes. Oh. Okay. Sure. Lisa, can I interrupt for just a quick moment? We just want to make sure that
Hear me
everyone
now.
can hear you well, and I know we're having a little bit of a
Much better.
low audio issue is all. If you
You're
could go
welcome.
to the
Thanks,
drop-down
Jennifer.
arrow at the top of the screen, we can try to
So in terms of the current knowledge of how homologous chromosomes are organized, it is controversial, right? Where there is one model, let me see here, I have my little mouse here, proposes that a pair of homologous chromosomes are aligned in this non-random anti-parallel configuration where they're at 100-degree angles from each other, right? But what has happened in subsequent reports have actually refuted this model and suggested that a pair of homologous chromosomes are randomly arranged relative to each other. So we've actually sought out to reconcile these conflicting studies to investigate the spatial relationship between a pair of homologous chromosomes. Okay, so to study the organization of homologous chromosomes within dividing cells, we first examined homologous chromosome organization at prometaphase by chromosome painting in green a pair of, let's see, where's my mouse? There it is. By painting, right, chromosome painting in green a pair of homologous chromosomes, right? So what you're looking at here is an image of a human cell at prometaphase with the DNA stained in gray. And what we did was we measured the 3D angles between a pair of these homologous chromosomes. And so as you can imagine, if there was this anti-parallel configuration, an optimal 180-degree angular relationship would be achieved. So what we did was we quantified this relationship using a Rose plot. And what we found was that there was no enrichment of homologs arranged at this 180-degree angle, right? So this is just a frequency distribution plot. It's a Rose plot where each of these wedges represent the number of cells that shows this angle relationship. And on the periphery, what you see is the 0 degrees, 30 degrees, 60, and so forth. And the bottom line is, at that 180-degree mark, we didn't see enrichment, right? So this demonstrated to us that, or actually, let me just keep going here, but although we didn't see this enrichment, what we did consistently see was that homologous chromosome pairs were consistently segregated from one another throughout mitosis. Okay, so then we wanted to determine if there was a fixed position or address for each of these homologous chromosomes. So what we did was we measured the, the minimal distance and also the angular relationship between the homologous chromosome pairs, right? And what we found was that, and we mapped it across different cell cycle stages here, but the main takeaway is, right, that we don't see any type of hotspot or fixed or defined distance or angular relationship between a pair of homologous chromosomes at any of these particular stages, right? However, and part of it was because this original idea that proposes a 180-degree relationship made an assumption that each of these chromosomes had a fixed position, right? So in our quantification down here, what we found was that there was an absence, right, that there was no hotspot or fixed position for each of these homologous chromosomes, and also, right, an absence of pairing between the homologous chromosomes, right? And that's when the distance and angular orientation would be at 0. So that asterisk represents, right, this absence of pairing between these homologous chromosomes. So essentially, our set of data told us two important things. One, homologous chromosome position is not fixed, right? And two, homologous chromosomes actually do not pair and get together. And so this supports an underlying mechanism to prevent homologous chromosomes pairing within a cell. So what we wanted to do next was to systematically map the positions of homologous chromosomes, right, by creating first an axial coordinate system. But before we could create this coordinate system, we had to understand the dynamics of chromosome behavior to determine how much displacement or rotation was present during mitosis. And so that is what we did next, right? So what we did was we employed a human cell line that stably expresses a SEMP GFP to visualize the chromosomes, and a centron GFP to basically visualize the centrosome, which helped us label the nuclear division axis. And what we did was we recorded these cells undergoing mitosis on a spinning disk microscope, right? 3D reconstructed each of these chromosomes, and then that allowed us to individually track the trajectory of each of these chromosomes throughout the cell cycle. Okay, and so Jennifer, this is where if you want to have that video, uh-oh, Jennifer? Yes. And so what you're basically looking at is, let's see, uh-oh. Oh no, I guess that video is not working. Oh, here we go. Okay, so you're basically watching a video of a cell undergoing prometaphase to metaphase. And so basically this video is showing you, so you're looking at, let me get my little pointer out. Okay, actually. You're basically looking at individual chromosomes. You see the legs wiggling around here. And what I did was each chromosome is labeled by these circular dots. And just for easier visualization, I pseudo-colored these dots so you could track them over time. And so you're basically looking at a chromosome or chromosome behavior from prometaphase to metaphase. And what you can appreciate from this video is that, you know, the centrosomes are basically undergoing this 90-degree rotation. So this told us that probably this was not the appropriate stage to, you know, basically construct a map or coordinate system. All right, Jennifer, we're going to go to the next video. Okay, so now what we did was look at a subsequent stage, which is from metaphase to anaphase. If you want to drag that here. Thank you. And so what you see in this video, right, is that these chromosomes are moving basically in parallel, right, to the mitotic spindle, showing these very stereotypical manners where you're seeing, again, this parallel movement relative to the spindle and not, or an absence of this perpendicular movement, right? So we essentially found that chromosomes displayed less rotation along the nuclear division or centrosome axis at this particular stage, metaphase to anaphase. Okay, Jennifer, next video. Last video. Thank you. And so I'm going to make this short, is then we looked at, finally, anaphase to telophase, and what we found, there we go, is that at this particular stage, which is very short, right, you're seeing a lot of rotation. Okay, so telling us that this was not an appropriate stage to analyze. So you don't have to worry about the video here, Jennifer, but essentially to summarize, what we found was the most appropriate stage to basically perform or do a coordinate analysis, right, to make a map was at metaphase to anaphase. So I'm just going to kind of wrap it up with just a couple of, oh, my light is going off. Okay, a couple of slides. Hold on. Okay, is how we essentially created our axial coordinate system was where we had the x-axis defined by the nuclear division plane or centrosome axis. Okay, let me get my mouse here. Okay, the z-axis is defined by the microscope or laser lines as perpendicular to the cover slip, and by definition, perpendicular to both the x and z-axis, we have the y-axis. Okay. And let's see. Okay, so what you're seeing here is a confocal stacked image of an anaphase human cell painted for chromosome 4. Okay, what we did was we used a 3D reconstruction program, okay, to basically render this into 3D volumes, right? And so essentially, when we overlaid many nuclei on top of each other, although the position of chromosome 4, let me get my mouse here, although the position of chromosome 4 was varied, what we found was a surprising and unique pattern that did emerge, right? We found that one chromosome, and not both, tended to stay on one side of the nuclear division plane, which essentially divides the nucleus into two halves, or what we like to define hemispheres. So I'm just going to probably end it on a few slides here because I'm running out of time, but what you're looking at is at one anaphase nucleus with a homologous chromosome pair. What we've done here in the overlay is we have one homologous chromosome labeled in green and its corresponding partner in white. And this is essentially an overlay of 30 nuclei on top of each other, or mitotic cells on top of each other. And it's very obvious, right? You have a green zone and you have a white zone. And essentially what we find is that there's this one homologous chromosome per side, right, in this particular zone. So So we really want to ask, and I just want to conclude here, is this one homologous chromosome per nuclear hemisphere, what we like to now define as an anti-pairing pattern for homologous chromosome, is that present for all the other chromosomes in the human karyotype, right? I'm showing chromosome 4, but is that present for all the autosomes? And the answer is yes, right? So I performed the analysis looking at the karyotype, and we find that they have the same shared motif where there is one homologous chromosome per nuclear half or nuclear hemisphere. And what was surprising was, even when we looked at the sex chromosomes in females and males, this pattern still was conserved. Coming to our main conclusion, that we found that one haploid chromosome set essentially forms one nuclear hemisphere, right? And so I'm not going to really go into or show any more videos because I'm running out of time. But we basically further characterize this in disease cells in cancer. Oh, okay, Jennifer, I'm just going to keep going, okay? And what we found was, previously there was this renal carcinoma cell that has been described to have abnormal gene transcription for genes on chromosome 19. So we want to really test whether there's a correlation with this abnormal gene transcription and this haploid chromosome set-based anti-pairing organization pattern. Okay, so I'm going to end it here. That we found that essentially when we did this test on these cancer cells, that the cancer cells do lose this one homologous per hemisphere pattern, and suggests that this pattern that we found may function to actually keep homologous chromosome pairs apart. So really in conclusion, right, this data shows that our one homologous chromosome per hemisphere pattern may prevent or minimize allelic misregulation and genetic recombination in complex genomes during mitosis. So essentially our model here is, and I've skipped a lot of slides, I apologize, was at prometaphase we see that the homologous chromosomes are already organized and segregated. And we think the haploid sets are present already. However, I have students now looking into how chromosomes are behaving as your cell transitions from prometaphase to metaphase. But that essentially this haploid set organization is present at metaphase, and we've documented this, and this pattern basically persists to anaphase. And so with that, I want to go ahead and end with my last slide and thank you all for your patience as I've been troubleshooting this presentation. And I want to acknowledge students in my lab. I am very fortunate to work at Sonoma State. I have not only great colleagues in my department that have been very supportive through my transition here, but also I work with many dedicated and fantastic students. And I want to thank especially a lot of my administrative help and a lot of other staff here at Sonoma State who have been nothing but so wonderful to me and helped support me as I started my lab out here. And so I also finally want to thank all my funding sources for their support as well. So thank you. And I'm sorry, I want to introduce Clinton Francis from Cal Poly SLO. Sorry.
Hi, thanks a lot. Thank you all for sticking around to the end of our webinar here. I too, I'm coming to you all from the future. I'm here in Europe where it also is night, like for Pleuni. And I'm currently on sabbatical from Cal Poly at doing a postdoc at the Max Planck Institute of Ornithology. So what I'd like to do is I'll get to a few tips for trying to maintain research programs at a CSU, and also try to just give you a general flavor for some of the types of projects that we do in my lab. We, at Cal Poly, we have a master's program, so I have a lot of different master's students that work on these projects. Also have supported postdoctoral associates in my lab as well, visiting scientists, and of course a lot of undergraduates like all the CSUs. So one of the things that we're really interested in is just trying to understand where species live and how they behave according to different sensory gradients in environmental conditions. And so some of the gradients that we're really interested in are variation in aspects of the environment that we don't typically think about or recognize as humans, such as, variation in light regimes. That could be from dark forests to really brightly lit meadows, or even changes in anthropogenic night lighting through lights and sky glow. We've also had a long time interest in changes in acoustic conditions, whether those are from, traffic and urbanization, or gradients in natural sounds, for example, going away from a coastline or being close to rivers or chorusing insects. So in all of our work, what we really try to do is try to adopt this idea of embracing each species' Umwelt. And that essentially is this idea that every single animal has its own unique window into reality. Every organism has different sensory strengths for perceiving the world. And if we really want to understand where species live and why they live in the places they do, or how they behave, or how they interact with one another, what we really have to do is recognize these sensory strengths and do our best to explicitly measure the dimensions of the environment that are important to these particular species. So in my lab, what we generally do is we take two different kinds of approaches. I started as a field biologist, but I had a postdoctoral position at the National Evolutionary Synthesis Center, which taught me to try to repurpose existing information and use a lot of big databases. And so we really adopt those two approaches in the lab. So in the field-placed types of experiments, we typically work with birds, but over the last 8 years or so, we've also worked with bats, various small mammals, insects, and even done experiments with people as well. So what these experiments tend to look like, they can be either small scale or quite large scale. Just to give you a flavor for what some of those might look like, we might have short-term modifications through playback of the environment to see how animals change their behavior, or manipulations to see how people actually change their behavior on natural areas and trails, such as Muir Woods. On larger scale projects, we've done things like simulate the sounds of oceans or rivers in areas where there are not oceans or rivers, and to try to understand how just changing these acoustics can restructure bird, bat, and insect communities. On the big data side of things, just a couple of example projects include using citizen science data from bird watching, either watching birds at feeders or monitoring their nests, to try to understand how fitness or abundance changes with all types of different aspects of human disturbance, including gradients in noise and light pollution. Another example of our work using big data is taking advantage of County Health Board sentinel chickens that monitor for arboroviruses. And so across Florida, we've taken advantage of these sentinel chickens to try to understand some of the drivers for West Nile virus emergence and how it actually relates to light pollution through a combination of changes to hosts in response to light pollution, but also changes in vectors and those mosquitoes being drawn to lights. So I'll just give you a flavor for some of the more recent work that my students and I have been doing at Cal Poly, and then try to get to some of our take-homes and some of the lessons that I've learned through trying to maintain a program at Cal Poly. So at our, we maintain a field site in New Mexico where we've been working for about 15 years. And one of our recent efforts has been that we've gone back and we've measured vegetation communities, specifically species that depend on animals to disperse their seeds for successful germination. And what we can see from these surveys is that the two foundational species, or the most important woodland species in this area, the pinyon pine and the Utah juniper, their recruitment declines quite strongly with increases in background noise from industrial activities. And we've been able to tie this to changes in the behavior of some of these key seed dispersers, whether they're seed caching rodents and birds that essentially hide the seeds of pinyon pine in little caches in the ground, or frugivorous animals such as foxes and bluebirds that ingest these juniper seeds and scatter them away from adult trees. Another example of a recent project that we've done was using this NestWatch data set from the Lab of Ornithology at Cornell's project, where we used over 58,000 nests to try to understand how fitness changes for birds in response to noise and light pollution. And so there's a lot of complicated results to this particular study, so I'll just boil it down to just a couple of interesting ones. But what we tended to find is that some of these nesting outcomes and even the timing of when birds actually nest tends to be tied to some of the traits. So for example, birds that have these really low frequency vocalizations that can essentially be masked or covered up by noise, they tend to nest much later in the season in response to noise pollution. In contrast, birds that have eye geometries where they can see really well under low light conditions, they tend to be really sensitive to any kind of intrusion from artificial light, and this causes them to advance their nesting quite substantially, sometimes even a whole month earlier than they would normally nest. And we've also shown that this figure on the right didn't come out right. It's supposed to be just one of those panels, but we've been able to tie some of these traits to actual fitness outcomes as well. Specifically, these light gathering ability of the eye also helps explain how species respond in terms of nest success to light pollution. And the main story there is that birds that have this dim light vision end up doing better with light exposure. And this has some really interesting implications for how these sensory pollutants interact with climate change. Another fun project where we've messed around with humans is we did what we call a phantom chorus, where visitors to natural areas walked through 2 different trails where we exposed them to birdsong through playback in hidden speakers. And the overall objective of this study was to try to see, like, do people tend to perceive elevated diversity of animals acoustically, and does it have an improvement in their overall well-being, self-reported well-being, assessed through a large survey with a whole bunch of different questions. The main message that we found is that the application of this phantom chorus does seem to improve people's self-reported well-being, and that this increase in their well-being was related to their perceptions of biodiversity in these areas, which suggests that even hearing birdsong is one of the modalities by which we get these psychological ecosystem services from interacting with nature and influencing our overall well-being. So some of our major lessons from this work and others so far is, these changes in noise and light regimes can have really drastic consequences for animals and their supporting ecological systems, from changes in behavior, phenology, all the way down to interactions that cascade down to species that don't necessarily have any clear link to the acoustic world, such as plants. And then finally, we also are seeing that these variation in natural sounds can also cause similar patterns as do these anthropogenic sources. So these are environmental conditions that birds have been dealing with and other animals have been dealing with for a very long time. And finally, these natural sounds seem to have pretty important links to how humans acquire benefits through psychological ecosystem services. Okay, so our next step in my lab is I've been getting really interested in these traits and how they help explain how animals are responding to these anthropogenic stressors. So one thing that we're hoping to start doing over the next several years is try to start understanding the biogeography of these sensory systems. This is important because, these light gathering ability of the eye is not well known for very many species. Hearing abilities of, say, songbirds or any birds, we know this information for maybe 60 species out of 10,000. So there's clearly a lot to learn about variation across species and also variation within species if there's selection at the population level. And then moving forward on this multisensory experience on the human dimension side, we're trying to understand, like, what combination of sensory stimuli seems to provide these greatest benefits to human well-being. And besides that, like how long do the effects from these experiences with nature help or persist in people who have had these experiences with nature? So finally, I'll just have a few comments about what I've learned across the years from trying to maintain a productive research program. I think at the CSUs, one of our most limiting resource really is time. And it can be really challenging to try to carve out the time that you need to conduct a lot of research, especially research that's going to be externally funded from NSF, NIH, NASA, a variety of other agencies. And so I think having these really frank conversations with the department chairs, deans, whoever makes the decisions for you about what it's going to take to actually write these proposals and what it takes to actually do this work is really important. And this goes along the lines with not trying to overcommit. In terms of proposals, I'll echo what a few others have said, is make sure you talk to program officers before submission of the proposals, and also after submission. You'll find out a lot of interesting things that just don't make it into those summary documents. Something that I've found really helpful for proposals is actually removing myself from campus and home and focused writing retreats with collaborators to work on these proposals. That's where the lion's share of those tend to get fleshed out for me. I also try to not fit in too much. I use a lot of figures and keep things fairly simple so that it's not too much of a burden for reviewers. The other thing, at least for NSF proposals, is there's 5 criteria that reviewers are supposed to assess you for, and so you might as well just provide them with a summary at the very beginning of the grant that tells them exactly how your project has met those criteria. And in doing so, reviewers are busy. They'll often just copy and paste that right into their summary statements, and it can help elevate those proposals to the top of the pile. And in terms of working with undergraduates, I've found that recruiting them early is really important for my lab. We rely on, a lot of complicated acoustic techniques, a lot of complicated modeling, and it just takes a lot of time to get them up to speed to be really productive members of the lab. And I can say that some of my undergraduate students that started as freshmen or sophomores ended up being even more productive than some of our master students because they had the know-how of how to do all of the different things that we do in our lab. And finally, I'll just summarize that we still have a lot to learn in ecology from adopting this sensory biology lab. And as we try to expand on some of these other fronts, if there's anyone out there that's interested in sensory systems and genomics, please talk to me. Just always looking for new social scientists to collaborate with on some of these projects. And I'm also looking for physiologists that might be interested in contributing to auditory evoked potential work. And with that, I'll thank all the students and collaborators that have contributed to this work, as well as a couple of different NSF grants, also from NASA and the National Park Service. And with that, I'll turn it back to our moderator, Frank Gomez.
Thank you, Clinton, and certainly everyone for your wonderful presentations. Your work is really fascinating and very, very exciting, and it's great that a lot of CSU undergrads and master's students are able to participate. We have a little bit of time for a few questions. I'm going to start off with one. This one will go to Kelly. It certainly sounds like you're doing a lot of mentoring and training at Long Beach. How have you been able to implement this? And is your training specific to faculty solely in biology, or are you kind of branching out to other disciplines in STEM?
Great. Thanks. That's a great question, Frank. I appreciate it. We have 2 programs that we've developed, and this is all part of the CSULB Build initiative. The first one is really research specific. It's STEM specific, not biology specific. And it's based on the Entering Mentoring program, and that's a hybrid, and we've already done work with, or done a Train the Trainer session with CSU San Bernardino, which has been really fun. The second one is we try to go more inclusive, and so it's actually all scholarly and creative activities, including research. So because there's so many things in mentoring that really cross the board, so things like managing microaggressions or mentoring for social justice, or how to actively listen as a mentor. And that one's fully online, and it's going to be completed by the end of spring semester, so by June 2021, and that will be disseminable to any CSU that's interested. So yeah, that's how we've been doing these trainings.
Great, thank you, Kelly. Our next question is for Gilberto. What are your views on taking probiotic supplements?
Yeah, thanks for that question, Frank. That's actually a very interesting question, because this is an area where the actual microbiome data doesn't actually match the patient's benefits, right? So people have looked at the studies of, if you take a probiotic, how does it impact your microbiome? And from the microbiome perspective, it doesn't really seem to change. In often cases, those microbes don't colonize and become a sustaining part of the community. But on the flip side, clinically, people that take probiotics do generally receive a benefit, at least a perceived benefit. So whether it's actual placebo effect or whether it's actually doing something that we haven't yet uncovered, I would support someone if they have the anecdotal data saying that they feel better, they have whatever symptoms they're trying to compensate and take care of. So the science is inconclusive whether it's actually changing the microbiome, but I think from most of the people that would take them would tell us that they do receive some benefit. So the science doesn't necessarily match the clinical data in the case of probiotics. I would also argue that in many cases we could just eat the right foods to select and support the right types of microbes. So I'm more, I guess, in the prebiotic camp, right? So the right types of fibers, the right types of foods to kind of support the right types of microbes.
Eat well and you'll do better, right?
Eat well and you'll do better, right?
Yeah, eat the right fibers. I mean, it's what we've been told since we were kids, right? Eat our vegetables.
Our next question is for Clinton. Related to your work involving noise pollution, is there any solid evidence that restrictions related to COVID-19 has influenced wildlife?
Sure. Yeah, thanks for that question, Frank. I think this is probably something that everybody can relate to. There were tons of news all over the media about how wildlife were suddenly invading cities all over the world with the shutdowns. I think a really important aspect of this is when we turned down the noise, a few orders of magnitude in terms of acoustic power. What we did at the same time is we essentially made our sensory worlds for what we can hear an order of magnitude larger. And so a lot of what is happening is people are just able to perceive a much larger world than they were under normal conditions. But there have been some pretty cool studies that have come out so far that have documented some of these changes. My postdoc, Jenny Phillips, had a paper in Science just a couple of months ago demonstrating that the shutdown in the San Francisco area has caused the species there, the white-crowned sparrow, to fundamentally change its song in a whole bunch of different ways that make sense from a sexual selection perspective. And I don't doubt there's going to be a lot more coming from people who have opportunistically taken advantage of this unprecedented opportunity.
Seen those videos on the news where we've seen animals kind of move into certain areas of near homes and in suburbs. So I can imagine that it's probably affecting probably all forms of wildlife. Yeah. So thank you,
Definitely.
Clinton.
Yeah. Thanks.
So given the time, we have one final question for Pleuni. We're probably all marveling at your video, and like I have mentioned in the chat box, I've seen it multiple times, and I'm still pretty marveled at it. Can you provide some of the information on the efforts at SF State to train biology and chemistry students in data science?
Yes, Thank you, Frank, and thanks for. I'm happy that you like the video. I like it too. You know, at SF State we have many different efforts to help our students learn data science, computer science, bioinformatics, and some of the things that we do are we now have a lot of computer science classes that are dedicated to biology and chemistry students, and we find that that helps them because we can focus on relevant topics for them, and they feel more supported when it's for them instead of a regular CS class. We also use mentors, other students that are paid to help our students in small groups, and then we also make a huge effort to be supportive and to have, you know, to support a growth mindset for our students so that they know that if they don't understand it immediately, they will learn. And it's paying off. We now have a large group of students, you know, like in my lab, in Scott's lab, and in many other labs who do data science projects and who are getting minors and certificates that will help them in their career.
Pleuni. And really thanks to everybody for participating today and in being here for the Q&A. Speaker contact information is provided here, or it will be provided very shortly. For information on any of the presentations, please contact the speakers, as likely they'd be interested in collaborating or potential proposal going forward. In addition, a link to the webcast will soon be available on the STEMnet website. We'll soon be following up to all the registrants with a survey. Your comments and feedback is very important so we can improve wherever we can in information that we provide you. For more information on this webcast or in previous webcasts, you can contact Monica Alacon, Administrative Analysis for STEMnet, or myself. We have a full schedule for next term, the spring term, or if you're at SLO, your winter and spring quarters, beginning February 4th with a CSU NSF IUSE program Exemplars, followed in March and April by a few other STEMnet webcasts. Registration information will be forthcoming in the new year. And finally, it's customary for me to say a few parting words after each webcast highlighting a current event. In the spirit of the holiday season, today will be a bit different. In the mood of giving thanks, I'd like to thank all the presenters who've presented in one of our 11 webcasts and 4 virtual research cafes this year. We could not have done it without you. Your willingness to present and share your work with your colleagues across the system is a testament to all. It's been a pleasure to work on collaborative proposals with faculty from 9 different CSU campuses. Your expertise, knowledge, and sheer drive has been truly inspirational. To those faculty involved with funded proposals, I look forward to long relationships going forward on working on these projects. And to those faculty on proposals in review, let's continue to cross our fingers to make sure that they are funded. Let's see, I think we have 5 NSF and 1 NIH proposal that we submitted over the past 3 months. I thank all the CSU presidents, provosts, deans, AVPs, and directors for research and staff and SPO offices for your openness and ability to work with us. By allowing easy access to your campuses and faculty, you've allowed us to connect to strengthen faculty collaborations in STEM disciplines as well as non-STEM disciplines, build research and educational capacity, and support faculty in developing competitive collaborative proposals. I thank Ganesh Raman for his excellent mentorship, taking me under his wing, and for being a friend and confidant over the past 14 months. I thank STEMNet's own administrative analyst, Monica Alacon, for being the greatest of help in the STEMnet office, albeit virtually since March, and for striving for excellence all the time. Lastly, I would be remiss if I did not thank executive producer Jennifer Wicks. She is a consummate professional and is the brains behind our high-quality webcast product. So we thank her a great deal for what we churn out every month or so. To all of you, be safe, have a great holiday season, and have a wonderful day. And I now turn it over to our own Jennifer Wicks for a few closing remarks.
Thank you very much, Frank. I really appreciate your kind words, and it's always such a pleasure to have the opportunity to work with you and to work with Monica as well on your team. She's an incredible asset as well. And I also want to thank Kayla Perez, my partner in crime, co-producer, for helping me at every turn on these webcasts as well. I'll go ahead and turn on my webcam here. The final webcast of 2020 here for STEMnet. So thank you all for your excellent presentations and your just your exemplary work, as is the title of this webcast today. Wish you all wonderful holidays and look forward to a whole new series of webcasts in the series next year. And wish you a great day, and we'll see you very soon on another STEMnet webcast. Thanks, everyone.
Do you want us to all stick around after, Jennifer?
Yes, that would be